Yeah. This is a brief overview of what I'll be talking about in the next 45-50 minutes. So just a brief understanding of the fundamental concepts of cancer in terms of molecular biology. And through different case scenarios, I'll be talking about the genetic testing in cancer, how to go about it, what are the complications, practical aspects of it in particular, pedigree analysis, and also understanding some of the basic genetic technical terms. So whether a cancer occurs in one individual in a family, which is sporadic because of a somatic mutation, or in some cases in many individuals in the same family as a hereditary trait, the one thing which is the fundamental aspect is that cancer is a genetic disease. And as true for all genetic disorders, or in fact most of the disorders that we know of, it is an interplay of the effects of the genetic effects and the environmental factors. So the balance of which one outweighs the other, or how much effect each factor has a role, that determines the outcome and the phenotype or the observable characteristics that we see in a particular disorder. So on the one hand, we have disorders which are caused by defects in one gene. That is, the effect is enough to produce the disorder. So those are pure or absolute single gene monogenic disorders, and these disorders follow the patterns or the principles of as described by Mendel, and hence it is known as, sorry. And these disorders are therefore known as Mendelian disorders. And on the other hand, we have scurvy, which is a purely environmental, which is caused by a purely environmental defect. But most of the disorders that we see are polygenic or multifactorial, where the effect is caused by many genes, interactions of many genes, and the modifying effect of the environmental factors, like for example our ischemic heart diseases, diabetes, etc. So if we look at the cancer, on one hand we have hereditary cancer syndromes which fall towards the Mendelian disorder, where the effect of a gene or a few genes is enough to cause the disease. But most of the cancers that we see are sporadic, and they are a combination of various genetic factors and environmental factors. So before going into the aspects, it is very important for us to understand these terms, which are germline mutations and somatic mutations. So germline, as the name suggests, this is pertaining to the gametes. So the mutation, that is the defect in the gene which occurs during gametogenesis in the parents. So if the parental gametes are mutated, or sometimes the mutation occurs in the zygote formation. So because of this, the cells in the whole body carry the mutation. So the mutation is present in all the cells of the organism, and hence in the subsequent generation, he passes on the mutation. Whereas on the other hand, we have somatic mutations. So somatic means cellular. So here we have normal gametes here. The embryo is formed normally because there is no mutation. However, after the zygote is formed, at various stages, a mutation can occur at a particular stage of development. So depending on the stage of development, the mutation to which organ it spreads, that determines where the phenotype or where the features are seen. So in terms of tumor, if one particular cell is mutated, the lineage, the cells which come from that particular original cell will carry the mutation, and hence they will manifest as a tumor. So this is This is, and all the other cells in the organism do not have the mutations. So this is only a mutation which is restricted to a particular organ or tissue, and hence it is not transmitted or into the next generation. So there are various genetic variations in a tumor. These may vary from hundreds to thousands. And if we look at the different types of tumor, most of these variations or mutations, as we call, they are random and not specific. Like, for example, if we look at breast cancer and some liver cancer, most of the mutated variations are in common genes. So these genes are not specific to a particular type of tumor, and these are better known as passenger mutations or passenger genes. So, but when we observe closely, there are some mutations which are recurrent and which are specific for a particular type of cancer, and they are presumed to cause or have an effect on the causation or progression of that particular tumor. So these are known as driver genes, and the mutations in these genes are driver mutations. So broadly and very simply, driver genes are proto-oncogenes and tumor suppressor genes. So this is in very simplified terms of the fundamental aspect or concept of cancer development. So if we look, most of the cancers that we see, they are sporadic. That means they are a single occurrence in families. Really sorry for the background noise. So but if we look at some families, we may find that there are a clustering of same type of cancer in different family members across different generations. So we are very much interested in this particular subset of families, because this is the starting point of investigating and understanding the molecular concepts of cancer evolution and development. So most of the sporadic cancers, as I had shown previously in that diagram depicting the genetic and the environmental factors, they lie somewhere in between, and they have both the genetic and environmental factors. And there are various studies which can determine or more appropriately try to estimate how much is the increased risk as pertaining to the presence of particular genotype, but these studies only tell us about the relative risk. They do not give us an absolute risk, and in most of the cases, the increase is just by some folds. But in 5% of cancer families, we have what is called the typical Mendelian inheritance, as observed in single gene or monogenic disorders. So these familial cancers are what is known as the hereditary cancer syndromes. So there's a long list of syndromes, some of which I have listed here.
If it is infiltrating,
Sorry, is someone saying something?
I request everybody to mute their audio. And Amita, sound is not very clear. Can you just increase your volume if it is possible?
Yes, sir. So is it better now, sir?
Now it's better. Yeah.
Okay. Okay, sir. So to understand, so most of the genes which are involved in cancer formation and progression, these are the tumor suppressor cells. So basic molecular biology, if we look into, then there is something called the Knutson's two-hit hypothesis, which was first described in retinoblastoma tumor cells. So here what happens is, on the one hand, we have hereditary cancer or familial cancer, and on the other hand, we have sporadic cancers. So in the hereditary cancer, because of the germline mutation, there is there is one copy of the gene. The cross mark here indicates a mutation, and these are the two chromosomes or two copies of the genes, it each inherited from one parent. So the cross mark here is in one chromosome or one gene only. So this is already mutated. One copy is already mutated from birth because of the germline mutation. As the cells divide, due to some random phenomenon, because as cells divide, there are chances of errors in DNA replication, errors in repair, which causes the mutation. So the second hit may occur anywhere in any of the cells. So when both the copies are mutated, then the tumor occurs. Whereas when we look at the sporadic tumors, the first, to begin with at birth, there is no mutation in any of the alleles. But because of some random phenomenon, in one of the cells, one of the alleles have a mutation. Then as the cells divide, the second normal copy may also be mutated with a second hit. So when both the alleles again are mutated, this again causes tumor. So from this diagram, it is evident that if we have an already baseline mutated allele, it is easier for the second hit to occur, and therefore the tumor to occur. But if we don't have a baseline germline mutation, the two hits occurring in the same gene and knocking out the cell, that is a less common, or it will take more time, and it is a sporadic occurrence. So when we look at the clinical presentation in terms of familial history and the clinical presentation in patients, we can have certain clues which point us towards what type of cancer we are looking at. It is sporadic, or is there a clue pointing towards Mendelian or familial cancer syndromes? So first is the family history or the family tree here. So here we have, I'll be explaining this in detail in the subsequent slides. So here we have different individuals in different generations affected, multiple individuals. And in sporadic, it is usually only one individual which is affected in the family. In familial cancer, there is already a germline mutation and the somatic mutation occurs, and therefore the chances are more that the tumors are multiple, it is bilateral, and it is early onset. And in sporadic cancers, both the mutations, both the hits have to occur during the lifetime of the individual. So it is usually later onset, it is a single tumor, and it is unilateral. So these are in very simplified terms, the characteristics of familial and sporadic cancer. So with this background, we will come towards more practical aspects which we face in the clinics. Most commonly, it is in terms of genetic testing. So if we speak about, talk about genetic testing in cancer, broadly, we have two categories. One is germline testing and another is tumor testing. So when we talk about genetic testing, most of us by default, we mean germline testing or testing in the blood to determine whether the individual already has a mutated gene from birth. So the samples which we collect mostly for germline testing is blood, since it is since we presume that the mutation is present in all the cells of the body, and blood and saliva are some of the samples which are easily available. And another is tumor genotyping. So I'll be talking mostly on germline testing in cancer and demonstrating some of the cases that we have faced in our clinics. So we have here the first case. So we have Mrs. B, who is a 45 years female, and she is the index case. So in terms of genetics, the term that we use for an index case is proband or sometimes propositors. So she has come to us with breast cancer diagnosis at the age of 35 years, and she has already undergone bilateral radical mastectomy. She has a 25 years old daughter who's healthy. So on the right hand side of your screen, I will be helping you draw the pedigree here. So circle means a female, and if it's shaded, it means there is some characteristic or some observable characteristic which is denoted by that particular color, which has to be explained in the legend given in the figure. So here we have B, age and diagnosis at what age, and this arrow with the P sign, this denotes that this is the proband on the index case. So she has a 25 years old daughter who is healthy. So this horizontal line is the mating line, and the vertical line is the line of the progeny. So her daughter, so this is this particular individual. So she is 25 years old. So a circle, and she is healthy. She is clinically healthy. So there is no color. We have not shaded her with any color. So this essentially means that she is clinically asymptomatic. Then next, so this Mrs. B has two healthy sisters and a sister who died of ovarian cancer. So this is the extension of the family tree. So this Mrs. B has two healthy sisters here and another sister who died of ovarian cancer. So here again it's shaded. So someone may ask why I have shaded it in the same color as the breast cancer. So here I'm taking that the breast and the ovarian cancer are because of the same cause. It depends, you may shade it in a different color. Then the slanted line here, which cuts across the circle, this denotes that this individual has died. So if we have further information, we should note here at what age she was diagnosed and when did she die. Then the circle are the females and the square is the male here that I forgot to add. So then this Mrs. B's mother, she died at 65 years of age due to complications of diabetes mellitus. So this is the mother. So again, we have another generation here. This is the mother who died at 65 years, and she died because of complications of diabetes. She did not have any history or documents which were suggesting that she had any tumor related issues. So she was shaded with a different color, denoting that this is something else. Then we have another maternal aunt who died from breast and ovarian cancer. So this is the aunt here. Aunt is maternal aunt here, who died from breast and ovarian cancer, and she has a son who has prostate cancer. So I hope everyone is following this. So it just needs some practice to draw this and understand it and make it comprehensive. Then we have B's maternal grandmother who also died of breast cancer. So this is the previous generation where Mrs. B's mother's mother, grandmother, she died again of breast cancer. So this is the 3 generation, or in fact, it is a 4 generation pedigree. So we denote the generations with the Roman numerals here, first, second, third, and fourth generation. And here we denote what each color means. So blue is this and so on. So this is the complete pedigree that we have drawn here. So analysis of the pedigree gives us certain information. So as seen in the previous case, many times we see such family trees where multiple generations are affected and males and females are almost equally affected, and the disease can be transmitted both by males and by females. So previously, when the causative genes were not identified, people used to study this large family trees for certain disorders, and they used to note common characteristics. So that is how they identified inheritance patterns. So when this type of inheritance pattern is observed, that is all multiple family members are affected on each generation, both males and females, then this denotes an autosomal dominant inheritance. Then subsequently, when the causative genes were identified, it was established that in such disorders, the mutation or the abnormality in one copy of the gene is enough to produce the disease. So that is what is known as a heterozygote. So here I have denoted the two copies of genes. So each one of us has two copies of each gene, one inherited from our mother and one from the father. So at a particular locus or at a particular location, if both the genotype is similar as here, then we call it homozygous. When it is different, then we call it heterozygous. So in autosomal dominant inheritance, the mutation in one allele only, that is a heterozygote, will manifest the disease. There is no need for both the alleles to be mutated. And when the gene lies in an autosome, if we look at the chromosomes, we have 23 pairs of chromosomes, where 22 are autosomes, which are same in males and females, and the 23rd pair is the sex chromosomes. So when the gene is in the autosome and it follows a dominant inheritance pattern, then such disorders are known as autosomal dominant disorders. So most of the hereditary cancer syndromes, they follow this particular pattern of inheritance. So coming back to the case for the hereditary breast cancer. So when such a family presents to us, the first question that we usually ask is genetic testing indicated? And here and in subsequent slides, genetic testing, unless specified, means germline testing. So first question that we ask is, should we go for a germline genetic testing in this family? So there are certain guidelines and clinical criteria which have been devised by various societies for each type of cancer to screen and to segregate families where genetic testing is more likely to yield a positive result. So it doesn't mean that if someone does not fall or fulfill this criteria, he or she might not have mutation in a hereditary cancer gene. It's just that with this criteria, we are more able to pick up those cases which are likely to have mutations in these genes. So as we get to know more and more about genetic etiology and mutations are identified, you must have realized that these criteria is extending and it's widening to include more cases. So that's particularly true for widely studied cancers like hereditary breast and ovarian cancer. So the particular family who visited us fulfills this criteria, and hence we decided to go for genetic testing. So the next question is, whom should we test in this family? So when we have a family with such presentation or for any genetic disease in general, we should always test the patient who has an unambiguous phenotype. That means we are very sure that this particular individual has the disease. So for example, if we are targeting hereditary breast ovarian cancer in this family, we will not like to go for this particular individual with prostate cancer, because this prostate cancer may be caused because of breast ovarian cancer gene, or it might be a sporadic occurrence due to some other genes. So we would like to target one of these individuals with proven breast cancer. So that is the first dictum here. Then once we have tested the individual and we have identified the causative variation, then subsequently we can test the family members who are at risk for that particular variation. So this is our usual approach. So then someone might ask, why can't we go directly for a direct testing in the asymptomatic individuals without prior testing of the affected patient. Like for example, this particular individual, why can't we test her directly? We can test her, it is possible, feasible. However, we will have certain limitations in interpreting the results. So that we will discuss in further slides. So the dictum is you should test the individual with the disease without any ambiguity. Then once we have decided whom to test, then the question which we face and which really, it's quite complicated is which test should we order. So again, these are certain points which will help us in deciding which test we order. So first is the clinical indication. Why are we testing? It depends on what is our provisional or clinical diagnosis, and whether we are testing it for diagnosis, confirmation, or whether we are testing for a pre-symptomatic or asymptomatic testing of at-risk individuals. So we should know our target. We should know the why we are testing, what we are testing, and the attributes of our weapon. That is what the test is able to detect and not able to detect. So for this, we need to have some background knowledge regarding what is the probable or what is the known genetic etiology of that particular diagnosis or that particular condition. So basically this consists of how many genes are known to cause that particular disease. It is only a single gene, or is it can be caused by multiple genes, and what are the type of mutations or variations which cause that particular cancer syndrome. So by types of causative variations, I mean, is there a point mutation, which is also otherwise known as single nucleotide variations, or is it large deletion or duplications, which are called as copy number variations? So I'll be explaining this in the next slide. And of course, availability of the test and the cost factor, which are the bottlenecks at present in our clinical practice. So these are some of the basic questions which need to be answered before choosing our test. So to understand which test we should choose, I'll go on to the next case here. We have this family where this 35-year-old man, he came to us with pheochromocytoma, bilateral and cerebellar and retinal hemangioblastomas, and a diagnosis of von Hippel-Lindau was made. Subsequently, family history, we could identify many affected or presumably affected, similarly affected family members, and they wanted to undergo genetic testing for management as well as pre-symptomatic testing of other family members. So von Hippel-Lindau or VHL, we all know is caused by one gene only, that is the VHL gene, and it is a very small gene which has only 3 exons. So exons, I hope everyone knows, the gene has coding part which are translated into proteins. These are the exons, and there are intervening sequences or the introns which are spliced out or which are discarded while translation or when it is made, converted into amino acids and protein. So it is a very small gene with only 3 exons, and the variant profile, if we look at available literature, it says that 90% of all the known mutations in VHL are caused by single nucleotide variations. So majority is because of point mutations. So in such cases, our test of choice would be what is known as Sanger sequencing, where each nucleotide of that gene is sequenced. So this is how it looks, the report or the electropherograms look like. So these are the nucleotides in each position, and where you see here, there is a double peak. There are two peaks overlapping. So that means the one of the alleles, one of the copies of the gene carries a different nucleotide change. So this is the nomenclature which is used: heterozygous, this is the transcript ID, and this is the gene name, and this is the DNA change. So this means that at this particular position, this instead of A or adenine, it is changed into G. So this will lead to a change in the amino acid at 73 position from glutamine to arginine. This is what it means. It's complicated and we need not understand, or we need not go into the details, but just a basic understanding. So I have used many technical terms in the previous few slides. So let's go back and understand some of the details or some of the basic information here. So locus, very simple, from location. So that means at a particular location, what is there, what gene is there, or what nucleotide is there. So that is the location. So here we have alleles. Alleles means alternate copies of a gene. So one gene, we have two copies, one from the mother and one from the father. So these are known as alleles. So if we look here at this locus A, the genotype, we write it as A small a. This is just for representation. In terms of the correct genetic terms, it will be a nucleotide sequence. So genotype, that means the genetic constitution of that particular individual at a particular locus. The effect or the result or the manifestation of the genotype, which is modified or which has additional environmental factors. So the ultimate result that we see is what is known as the phenotype. So these are the observable characteristics. And in terms of what we see in medical practice, it is the clinical presentation in terms of history, the signs and symptoms, or if we look at the tumors, even the histological patterns that are characteristic for each tumor, that is also a phenotype of that particular tumor. So then homozygous, heterozygous, I think I've already explained. So if it is similar in both the alleles, then it is homozygous. If it's different, it is heterozygous. So homozygous doesn't mean that it is mutation. Homozygous can be of wild type. That means it is of the normal genetic constitution is there in both the alleles. So it is homozygous wild type or homozygous mutated type, where both the alleles are mutated. Then types of variations or the types of mutations. So earlier we used to use the term mutations. Still today some of us do, but the term which is now preferred is variants or variations. So broadly, these are of two types: small scale variations and large scale variations. So small scale variations can be single nucleotide variations or small indels. Indels means insertions and deletions. So just for an example here, so this is the reference sequence. So reference sequence means the sequence of a normal individual, a control individual. So single nucleotide variation, if we look here at this, it is matching everywhere except here, where instead of T in the reference, in one of the alleles, it is converted to C. So there is a change in the nucleotide at this particular position. Then similarly here, there is an insertion of G and deletion of TT here. So these are examples of point mutations, or what is known as single nucleotide variations, and small insertions and deletions. So what is the effect? What are the effects of these changes in the genomic DNA? So the effect will be visible in terms of the protein, in terms of the function or structure. So at the DNA level, if there is a change, sometimes it might not change anything in terms of protein. These are silent mutations. Some may cause a stop mutation, some may cause a different amino acid to be changed instead of the original amino acid. So these are known as missense variations. Then insertions and deletions, if we look here, they will lead to the shifting of the reading frame, and hence they will encounter a stop codon prematurely and truncation of the protein. So these are the usual effects that are observed because of these various mutations. Then large scale variations. Many of us might have heard the term copy number variations, which is very commonly seen in cancers, in tumor tissues. So these are nothing but, if we look at the nucleotide sequence, we can just imagine here one alphabet each represents a segment of the DNA. So instead of one copy of F and G, this particular individual has 3 copies of these genes. So the copy number is variant from the original, or the, sorry. Okay, so the copy number is changed from the reference individual, and there is a triplication of the original segment here. So which is again shown by the below diagram. So these are some of the basic concepts for understanding. So with this, if we remember the previous VHL, so if no mutation is found on sequencing or Sanger sequencing, which constitutes about 90% of the known mutations in VHL, then we have to use another technique which can detect the large deletions and duplications, which are responsible in 10% of the cases. So one of the techniques that we use in our lab is MLPA, or sometimes we can use microarray also. So this particular individual has a deletion of exon 3 of the VHL gene. So the whole exon is deleted. To further understand some other concepts, we move on to family 3. So this is a family who has come with this particular individual here, this 33-year-old man has been diagnosed with medullary carcinoma of the thyroid, and his mother and his father had a history of same thyroid carcinoma and some ambiguous history or regarding intra-abdominal tumor, and she died in her 50s. So we don't know actually what the tumor etc is. So the physician or the endocrinologist had made a probable diagnosis of MEN2A in this family, and now we have to choose what testing we should do in the family. So the MEN2A is caused by the red gene, and red gene is a very large gene which has around 20 exons. So, but there are some locations or some mutations in the gene which are seen recurrently. So these are known as common mutations or hotspots. So 90% of the mutations in red gene pertaining to medullary thyroid cancer or MEN2A are concentrated in exons 10 and 11 of this red gene. So here again, we can use the same technique of Sanger sequencing and target only these exons. So most of 90% of the cases we will be able to identify the mutation. In the rest, if we don't identify, then we can go for another second tier testing with an alternative technology. So this is again the same variation with heterozygous variant in red gene, which is shown here. So in this particular family, once the variation or the pathogenic or causative variation has been identified, very important aspect is the pre-symptomatic testing of children. So in genetic testing, as pertaining to genetic disorders, there is a recommendation which says that children should not be tested unless they attain the age of majority. There are some exceptions, and one of the most notable exceptions is such families with medullary thyroid carcinoma, where an early intervention is life-changing. So definitely pre-symptomatic testing of children is recommended with subsequent management in terms of thyroidectomy. And even the timing of the surgery, it is now established that the genotype, that means the particular mutation which is found in that family, may help in determining when the thyroid, the surgery has to be done. So the American, I think the American Thyroid Society guidelines, they have classified the mutations in red gene into different classes. So some mutations are which are in higher class, like class D, they warrant an earlier intervention as compared to some of the others down in these lower down classes. So as we know, medullary MEN2A also has a pheochromocytoma or adrenal tumor as one of its manifestations. So someone may ask, why are we not advising or recommending prophylactic adrenalectomy for these children while we are doing thyroid surgery? So the answer to this we'll discuss in the upcoming slides. So coming back to the hereditary breast cancer family, which testing should be ordered in this family? Again, we go back to our literature search and we find that the most common genes responsible for hereditary breast cancer are the BRCA1 and BRCA2 genes. However, there are many other genes which have now been identified and associated with breast cancer. And this, even if we target only the BRCA1 and BRCA2 genes, they are very large genes with many multiple exons. So the same technique as was used in VHL and RED, can we use that? That is Sanger sequencing. Of course, we can use that. However, it will be not cost effective and it will be laborious. It will take time, reagents, etc. So this was done in in previous years when a better technology was not available. However, now the preferred method of testing in such cases is next generation sequencing. So what is the difference here? In Sanger sequencing, we sequence each nucleotide from one end to other end of that particular fragment. In next generation sequencing, the whole DNA of the gene, the whole DNA of the body of the cell. So we have estimated about 6 billion nucleotides in a particular cell. So we chop up the long strand of DNA which has all these nucleotides into different thousands of fragments and sequence each one simultaneously. So there is a massively parallel sequencing which is occurring. So this is now the preferred testing for most of the monogenic or otherwise also any genetic etiology to be identified. So there are different NGS based strategies which are now being very much marketed. So just a very brief, simplified explanation of these strategies here. So whole genome sequencing, we sequence the whole genome, including all the genes, all the 20,000 or something genes in the body, along with the coding, non-coding, intergenic sequences, etc. Everything has to be sequenced. So that is the whole genome sequencing. Then we have something which is known as whole exome sequencing, where only the coding regions or the exons of the genes. Here, if we look at the diagram, the colored parts are the coding regions and the shaded regions, these are the known coding regions. So in exome, we extract the coding parts of the genes and sequence only those parts. So this comprises only about 1 to 2% of the whole genome. However, these are the most important, functionally important regions of the gene and account for around 80 to 85% of mutations in almost all genetic disorders, monogenic disorders. So that's why cutting the technical complications and the cost, this is of maximum benefit. So this is whole exome sequencing. Then what we have something known as the NGS panel, which is very popular in terms of hereditary cancers. So panel means it's just a group of genes which are grouped as per your target of interest. So if you want to study the genes associated with breast cancer, then you may club together the genes associated with breast cancer and only sequence those genes. So this is again a very small portion of the whole. So what to choose amongst this depends on what you want, what your target is, and what is the availability. And but for cancers, what most of us prefer at this present point of time is to go for a panel sequencing, because our targets are restricted, and we do not know the consequences, or we will not know how to interpret the mutations in most of the other genes where association is not well established. So one must have realized that if we sequence the whole genome or the whole exome, then there is less likely chances for us to miss the mutations, since we are covering a larger area. However, if we are covering a larger area, then there are also chances of more data being interpreted. We don't know the interpretation of those variations, so it will create more confusion, and chances of landing with a variant of uncertain significance is more in such untargeted strategies. So most of us but in terms of hereditary cancer approaches, we are restricting ourselves to using NGS panels. But one thing we should, before ordering, we should ask the lab whether all the genes in that particular panel, all the genes causing the particular disorder are included in that panel. So that is a checkpoint which we need to be careful about. So in this, coming back to the first case here, sorry. So once we have decided that we are testing this individual, Mrs. B, for germline testing, then we have chosen BRCA1 and BRCA2 sequencing by next NGS. So they have come with this result. So many of us must be familiar with such kind of result here. So the labs usually give us the details of the variation which they have identified. So here is the name of the gene with the transcript, the location, the variation details, what is the protein change, what is the DNA change, what is the zygosity, inheritance, and classification. So as per the guidelines, there are classes of variants which need to be classified as with different criteria which are used. And pathogenic means it doesn't equate to being causative. It just denotes that the probability of the particular variant being the cause of disease is high. It does not equate causation. So sometimes we might not get such a result. We might get that there is no pathogenic or likely pathogenic variant detected. So these are what we commonly call as negative genetic test report. So does it mean that the individual does not have any genetic predisposition? No, it is more likely that the technique used for testing has several limitations or pitfalls, due to which it is not able to pick up that mutation. So the possible reasons might be the target or the mutation is not covered, is not sequenced properly. So again, we have to go back and see which genes are being covered. Are being covered in the NGS panel. Then NGS at present cannot detect the large insertions and deletions. So for this, we need to use an alternative method. Then sometimes there may be misinterpretation of the variations because of various algorithm abnormalities, errors in the software, etc. Sometimes the genes might be novel. Sometimes the mutations might not be germline. That means it is not an inherited mutation, but rather a somatic mutation. So we'll not find a mutation in the blood sample which we have used. So these are various reasons for getting a genetic report where it fails to identify the mutation or the causative variation. So I would prefer to use the term an uninformative genetic test report rather than a negative test report. And in such cases, we should always remember that there is a background risk. So even if there is no genetic predisposition, there is always a background risk of the individual getting or developing cancer, which is like 12% in terms of breast cancer. So this has to be explained to the individual or the family who comes to you for counseling. So coming back again to the same case, some complicated questions here. So why is the individual here, the individual shaded with green who died of diabetes complications, why didn't she have any features of breast cancer? She died at 65 years. So 65 years is quite a long time to develop breast cancer. So why didn't she have? Because her mother had and her daughter had. So she has to have the cancer to be able to transmit the mutation from her mother to her daughter. So why is she not affected? So that is the question which I'll answer in the next slide. Before that, let's again there's another question here where this particular individual C wants to know her risk and get herself tested. So she comes to your clinic and she gives this history, family history, and she says, I want to know what is my risk of developing cancer and I want to get myself tested. So how do we approach that? So basically two ways to approach it. First is, as always, we ask her to bring this particular individual who is alive and is affected so that we can test her, and then subsequently we can test for the same in her. So that is the preferred and the recommended way of testing such families. However, if it's not possible, suppose she is not ready to get herself tested, or she is no longer alive, etc. In those circumstances, we may go for direct testing of this asymptomatic individual. However, there are certain limitations. First of all, it will be an indirect interpretation. It will be an indirect conclusion of the causation of the mutation. We will not have a means to clarify or confirm that mutation had caused the breast cancer in her mother and other affected relatives. Suppose if we get a report which gives you a mutation in a gene already very well characterized, very well known, then we can be quite sure that that is the cause. But we cannot be, there's always some room for misinterpretation and error. Then more importantly, if we get a negative report or an uninformative report in this individual after testing, then we cannot conclude whether she doesn't have the risk, or else if she has the risk and we are not able to identify that risk. For example, if we go for an NG direct testing in an asymptomatic individual without a prior report available in the affected patient. So if we were to directly test this individual for breast cancer genes by next generation sequencing, and if her report comes out to be an uninformative report, then we cannot conclude whether she doesn't have the mutation or whether she has a mutation which has not been detected by the assay, because large deletions and duplications in genes are not detected at present by next generation sequencing. So it is basically the limitation of the technique which we are using rather than of the of the scientific basis or the underlying etiology. So we need to understand the limitations of what the test can detect or cannot detect, and accordingly counsel the family. before testing. So my question was, why was this particular individual here who died at 65 years did not show any features of breast cancer? So there is something which is known as penetrance. So in disorders with autosomal dominant inheritance, we might find such things where one individual who has the mutation doesn't manifest the phenotype, doesn't show the features. So this is observed as a skipping of generation in the pedigree. And this individual is said to have reduced penetrance or incomplete penetrance. So penetrance is, in terms of definition, it is the proportion of cases who have the genotype but who do not express the phenotype. So as we must have realized, penetrance depends upon the age of examination. So if we were to examine an individual at 15 or 16 years of age, by that time she might not have developed any symptoms or any signs of breast cancer, as opposed to examining her at 50, 60 years of age. So that is the first parameter, and it also depends on what measurement we are using to diagnose or label it as affected. So if we are using a clinical examination and appearance of a lump, etc., to diagnose breast cancer, then the detection will be at a later age, as opposed to using something like mammography to as a diagnostic parameter. So it is variable. And for different disorders, there are different penetrance. So all of the most or majority of the hereditary cancer syndromes, they do not have 100% penetrance. That means all of them show some amount of reduced penetrance in variable magnitude. So for BRCA1 and BRCA2 related hereditary breast ovarian cancer, it has been observed that there is a penetrance by 70 years of age of around 50 to 70%. So that means if we have 100 individuals with a BRCA1 mutation or a BRCA2 mutation and to follow them up till 70 years of age, then only 50 or 70 out of the 100, 50 to 70 out of the 100 will have manifestations of breast cancer. So that means even if this factor has to be kept in mind while doing asymptomatic or pre-symptomatic testing and subsequent counseling. So that brings to the previous question of MEN2A, where we recommended prophylactic thyroidectomy in individuals or in children who carry the mutation, but not adrenalectomy. So again, the answer lies in reduced penetrance for MEN2A. The penetrance of thyroid carcinoma is 100%, but for pheochromocytoma is less than 50%. So that's why adrenalectomy is not recommended. Risk of recurrence, again, easy to calculate in terms of the segregation of gametes. So if we have a dominant disorder, then the risk of recurrence is 50%, as denoted by this square. So all these red colored individuals will be affected and the black ones will not be affected. So out of the 4 possible combinations, 2 will be affected. That gives a risk of 50%. However, when we determine the risk of recurrence, we should always take into account the penetrance. So the last case here, this is a family who came recently to us. This 30-year-old was, she was the consultant, asymptomatic, and her father was diagnosed with parathyroid tumor, and the endocrine surgery who operated, he had suspected MEN1, and to confirm it, he had already sent a next generation based testing. So this was the report with which she came to us. And she wanted to know whether she has an increased risk of developing the cancer and what can be done. So again, MEN1 pathogenic variant was identified in the father. So again, the same principle, without testing, she will have a 50-50% chance of having the disease, not having the disease. So this 50-50 is just a figure or a statistic which does not have any significance in terms of an individual or his management. So to know in which half she falls, we need to test her. So testing of at-risk relatives. So again, we only target the particular mutation which was identified in the father. So we go for a Sanger sequencing. So in the daughter, the variant was not detected. In her sister, who had a parathyroid nodule and was yet to be diagnosed with MEN2, she harbored the variant, the pathogenic variant. And the brother, where we thought insulinoma could also be one of the manifestations of MEN1, he did not have the variant. So we can reassure her that she is not at increased risk of developing MEN1 associated cancer. This does not mean that she has no risk. The background risk will always be there. So this has to be conveyed to the family. So the sister who was diagnosed, again, she can be appropriately managed. And of course, the brother has some other cause of insulinoma, which could be a sporadic or somatic mutation. So this is the importance of family testing. Pre and post-test counseling is very, very important in terms of explaining to the family what is the test that we are ordering or being offered, what are the benefits of doing the test, what are the alternatives. Will testing help in management, formulating management or change in management plans? And we also should tell that the option of not testing, how it will impact and whether or how it will impact, and the expected results of the test, like a conclusive pathogenic mutation variant being identified, or an uninformative negative report and a variant of uncertain significance. Secondary findings, since we are testing by NGS a broad spectrum, then we might find a variation in a gene for a cardiomyopathy or a cardiac arrhythmia gene when we are testing for cancer. So it is unrelated to cancer, but still it has clinical significance. And of course, the limitations of the particular test, the cost, and need for subsequent segregation testing in the family. This is very important because most of the clinicians think that testing in the particular one individual and we got the result. But to confirm that result and to better classify that result in terms of pathogenicity, we will need to get the test done in affected as well as unaffected individuals in the family for to have more confidence in interpreting it as causative. Genetic testing of tumors, very limited experience we have. So it is the composite genetic aberrations or mutations in the particular tumor tissue. Various techniques, all genetic techniques can be used to identify. There are some customized panels which are already available, like mama print, which are very commonly used in terms of breast cancer. Then RNA profiling, expression profiling of tumors. This is another very upcoming and commonly used technique of classifying tumors and in terms of prognostication. So what is the utility of doing genetic testing of tumor tissue? So in terms of establishing a diagnosis, what we traditionally rely on is the histopathology, or sometimes the immunohistochemistry examination, which is not always reliable and has certain limitations. So genetic testing may help to better classify the tumor type, and it may help to prognosticate better and in terms of responses to various therapy regimens. And of course, the most important impact that it has is on in terms of targeted therapy, where molecules are available which target the particular mutation and hence has significant results in these tumors. So future, or I would rather say this is already in practice in some of the Western countries, liquid biopsy where from the blood we examine the DNA from the tumor instead of doing a biopsy and testing the tumor tissue, and personalized therapy where according to the genetic constitution of the individual, a tailor-made therapy strategy is devised for better efficacy and less side effects. So that's all from my side. Thank you very much for